liquid biopsies. In a moment, I'm going to hand over to Rafal, who will say focused on saliva-based liquid biopsies. In a moment, I'm going to hand over to Rafal, who will say a few words and introduce today's speakers. Before I do that, a few quick things. Questions. Feel free to ask our speakers questions whenever you like, using the area on the right-hand side of your screen. You can also upvote other people's questions. We should have plenty of time for Q&A at the end, when we'll bring all speakers back on screen to answer your questions. You can also use the chat section if you need any help with anything, and I will answer as best as I can. But questions for speakers should go in the questions area. On demand. If you miss something one of our speakers said, or if you have to leave early, the webinar is available on demand, so you can watch it back whenever you like. Just use the link in your registration email or go to our website at frontlinegenomics.com. The on-demand version becomes available just seconds after this live broadcast ends. If you enjoy the webinar today and you want one of your colleagues to watch as well, they can still register to see the on-demand version. Okay, back to today's webinar. Over to you, Rafal.
Welcome. DNA Genotek is excited to be hosting today's webinar, Exploring RNA in Saliva-Based Liquid Biopsies, a New Frontier in Precision Diagnostics. Today, we will hear from three distinguished scientists, Dr. Frank Middleton, Dr. Forrest Wright, and Dr. Andrew Spiers. My name is Rafal Iwasiow. I am the Vice President of Innovation and Technology at DNA Genotek. In today's webinar, we will hear how these two devices, the OmniGene Oral and OraCollect RNA, designed to collect saliva, are being used to drive patient experience and enable novel diagnostics. Both of these devices facilitate non-invasive sample collection and, through unique non-toxic chemistry, stabilize the sample for analysis in a laboratory setting. First, a little about saliva. Saliva contains a mixture of white blood cells, epithelial cells, and bacteria. We know that up to 74% of DNA in saliva is derived from white blood cells. Through the innovative collection device and chemistries, our solutions minimize the bacterial content. Here is an example of cellular morphology in oral samples. On the left, a saliva sample collected using OmniGene Oral collection device. On the right, using the OraCollect RNA device. In both, we see epithelial cells, lymphocytes, and monocytes. Saliva is a window into the human immune system and into systemic diseases. Saliva is an alternate sample type for liquid biopsy applications. Saliva is a great source of DNA and RNA, including small, non-coding RNAs. We know we can recover high-quality DNA from saliva, but we can also recover RNA, which includes mRNA and small non-coding RNAs, including microRNAs, which we know play important biological functions. These can be released by the local cells, but also transported through the body using, for example, exosomes. With that, I'd like to introduce our first two speakers. Dr. Frank Middleton is a professor in the departments of Neuroscience and Physiology, Biochemistry, Psychiatry, and Paediatrics at SUNY Upstate Medical University. He is the founder and director of the SUNY Molecular Analysis Core Facility, which specializes in next-generation sequencing analysis. Dr. Middleton is a highly experienced molecular neurobiologist, data analyst, and medical educator with a passion for brain research. His research pursuits using high-throughput genetic, epigenetic, genomic, and metagenomic approaches to neurodevelopmental and neurodegenerative disorders have led to the scientific discoveries about the role of RNA plays in the human body, including the early diagnosis of autism spectrum disorder, Parkinson's disease, and mild traumatic brain injury. Dr. Middleton's research revealed that particular RNA and microRNA related to autism, Parkinson, and mild traumatic brain injury were made in brain cells and found in saliva. This is one of the key reasons why saliva is providing a window into brain function. Next, I'd like to introduce Dr. Forrest Wright, who is the Director of Assay Development at Quadrant Biosciences. He earned his PhD from Upstate Medical University and has managed multiple research and development teams operating across the fields of cellular and molecular biology. As the Director of Assay Development at Quadrant Biosciences, Dr. Wright leads his team in the development of novel saliva-based assays in the fields of genetics, epigenetics, and virology, with the primary focus on neurodevelopmental and neurodegenerative diseases and disorders. These comprehensive testing solutions will utilize at-home saliva-based sample collection, ideal for telehealth-enabled practices, and allow for increased access to much-needed patient testing. With that, I turn it over to Drs. Middleton and Wright.
Greetings. I'm Dr. Frank Middleton from SUNY Upstate Medical University, and it's my pleasure to be able to describe our recent successes in harnessing the power of saliva for the development of diagnostic aids of brain disorders and infectious diseases. I'll start by pointing out that all of the work I'll describe has been done in collaboration with Quadrant Biosciences, which is based on our medical school campus. As this slide points out, spit is it. It all starts with saliva. That and a willingness to embrace our ignorance, recognize what we don't know, and allow cutting-edge technology and analytical approaches to accelerate the pace of discovery. The goal of our work has always been the same. We strive to make the most sensitive and specific biomarkers that could be of the most use in the early detection and enable early intervention to improve people's health and the quality of their lives. Our focus for today will be on highlights from our work in autism, Parkinson's disease, and COVID-19, though we've also achieved considerable success in concussion. So let's begin with a look at autism spectrum disorder, or ASD. Many of you are probably aware of the fact that ASD typically presents around age 2 or 3 as a constellation of somewhat variable symptoms that impact the way a child communicates and interacts with others, and also tends to result in patterns of repetitive and restricted behavior and an intolerance to change. This greatly reduces the ability of many persons with ASD to live independently or develop fulfilling interpersonal relationships. Several large-scale studies have indicated that the prevalence of ASD has continued to rise at a surprising rate over the past 3 decades, with current estimates in the US reaching approximately 1 in 44 children, or about 2.8%, with the prevalence being 4 times higher for boys than girls. And these trends, as this slide illustrates, are evident regardless of race or ethnicity. Ordinarily, this type of strong uniform increase would make some suspect that there could be a shared risk factor involved. And a lot of people spend a lot of time and effort looking at genetics for possible answers, given that ASD has been established as the single most heritable neuropsychiatric condition. However, despite all the effort that's been undertaken across several decades, genes by themselves have not yielded all the answers. In fact, even if you add up all the known syndromic causes of ASD, which are fairly uncommon, and all the strongest, more common genetic risk factors, you're still only able to account for about 10% of the cases. So something else must be involved. Perhaps something related to genes, but also responsive to the environment, or epigenetic in nature. So where did we look for answers? Approximately 10 years ago, we began to explore the potential for 2 classes of epigenetic molecules to play a role in ASD. These include microRNAs and Peewee interacting RNAs, or piRNAs. There are thousands of these in the genome, as this slide illustrates, and each one of these plays a role in silencing elements of the genome. MicroRNAs act at the level of silencing the translation of RNA into protein, and piRNAs, or Peewee interacting RNAs, actually act at the step before that, and they tend to keep the repetitive elements in our genome from being expressed into RNA. Now, importantly, these classes of non-coding RNA are not retained only in the cells that make them, but they can actually be released in exosomes and other carrier proteins where they can be transported in biofluids throughout the body. Those biofluid-carried microRNAs are bioactive, and they can play a role in changing gene expression in any target cell that they're taken up in. So we chose to look at these non-coding RNAs in saliva. Now, one question you may be asking is, why would saliva provide insight about a brain condition like ASD? And a simple answer to that is there are 5 pairs of cranial nerves that target the mouth that send signals to and from many of the target tissues in the oral cavity. This slide illustrates the nerves that go to the major salivary glands as well as the tongue in the mouth. Now, these nerve paths are the same ones taken by other RNAs, such as the rabies virus. When a person is able to spread rabies virus, that's because it has passed from the central nervous system into the peripheral nerves and ends up in the salivary glands. It's not because they've spread it from the site where it was inoculated first, when they were bitten. So RNAs, such as viruses, and the non-coding RNAs can be readily transported down these nerve paths into the oral cavity, where they may end up in saliva. And we've done a little bit of initial background work to establish the premise for this even further. That work was informed by work in protein, where we actually purified exosomes and examined the contents to see if there were neural-specific proteins that were actually expressed in these exosomes that we derived from the oral cavity. And in the upper left of this slide, you can see a picture of an exosome isolated from a normal, healthy adult person. Then you can see the size fractionation in the middle upper part of the slide for the population of exosomes that may have been purified from that individual. And most of the exosomes carrying microRNAs will be about 120 nanometers in size. When we look at the proteins that are expressed, we did it 2 different ways. In the lower left, you see an immunoblot for neural-specific proteins, which you can see are expressed at a very high level. Those are the ones that have red stars in the lower part of the panel. These levels of expression of those specific proteins exceed some of the more common pan-exosomal markers that are represented by the histobars in the upper part of that panel. There also are some glial-specific markers as well, indicated by the green star. In addition to these immunoblots, we also did shotgun mass spec analysis. And I won't describe to you the results for individual proteins, but when we looked at the functions that are overrepresented in the proteins that are most abundant by mass spec analysis, we interestingly found that Cajal-Ritzia cells and fetal brain cortex were overrepresented in the protein content that you find in normal adult humans. Now, the Cajal-Ritzia cells node that you see enriched with proteins from saliva is quite interesting because it's known to be implicated in early brain development. And these specific cells have actually been thought to potentially contribute to the pathophysiology of neuropsychiatric conditions, including autism. And there's a picture of these Cajal-Ritzia cells drawn by Ramon Cajal more than a century ago. In addition to the non-coding microRNAs, one of the things that saliva provides a window into is immune function. And one of the ways it does that is because it expresses many immune molecules. So another thing that we learned from our shotgun mass spec sequencing of protein content in saliva is that approximately 10% of the proteins in a normal uninfected adult person are antibodies. Most of those antibodies, as you can see in the pie chart in the lower left, are actually IgA class, but all the other antibody classes are represented as well. There are also other molecules that play a role in immune defense. And there's considerable data suggesting that children and adults with ASD have altered gut-brain activity, changes in their immune system, and therefore the microbiome came very much under our radar as something we'd want to look at closely in the saliva that we were collecting. So just to take you to the absolute milestone achievement that we were able to derive from all of this work involving approximately 1,000 children who were recruited into the study, is that we were able to combine the human non-coding microRNA, the microbial content, so the oral microbiome and the species that are represented there, and derive a classifier that was able to correctly predict in a holdout set more than 85% of the children. And it achieved a positive predictive value of 91%. And you can see in the upper right the collection device that was used for this large multi-year, multi-site study. It was actually the DNA Genotek oral swab that performed so well. So again, I want to emphasize that this performance was achieved in a holdout set that was not used to train the diagnostic model. I'd also point out that the best model that we were able to derive from the next-gen sequencing of the oral content ends up being about half microbiome content and half human non-coding RNA. Now, how does this performance compare to some other tests that you may be more familiar with? The behavioral analysis of autism is established a gold standard measure called the ADOS, or the Autism Diagnostic Observational Scale. And you can see that the sensitivity and specificity of the saliva-based test that we developed is actually higher than what's achieved through a purely behavioral analysis. Now, if you look at some other tests for infectious diseases like the flu or strep, or even a PSA test, you see that the saliva-based autism test performs very comparably or better than those. So we were very pleased with this outcome in the autism spectrum disorder research. Now, I would like to put the focus on a different part of the lifespan now, and that's looking at Parkinson's disease. So similar to the studies with autism, we recruited several hundred subjects for a study of saliva content in early stage Parkinson's disease. And we used subjects who were healthy, neurologically normal controls, as well as controls who had some condition, but not Parkinson's disease. And we tried to develop classifiers that would accurately predict the diagnosis in this case. Now, there's additional rationale for focusing on saliva in Parkinson's disease. And one of those is that most subjects with Parkinson's disease have difficulty in speech or swallowing. It's also the case that the pathology of Parkinson's disease, alpha-synuclein inclusions, which form Lewy bodies, has been shown to be present in salivary glands. Of most living subjects who participated in a biopsy study, as well as deceased subjects. And certainly the microbiome changes in the gut are now well documented in Parkinson's. And it's thought that the gut changes precede the motor changes you see by several years. So we were very excited to do biomarker development in Parkinson's disease. The functional assessment was very important to establish, and we used all of the standard questionnaires and functional measures that were the most appropriate to truly classify these subjects as early stage Parkinson's disease. And again, we focused on the host microRNA as well as the microbiome. Just looking at differences in expression on the left side of this plot, you can see what's a traditional volcano plot showing the corrected p-value compared to the change in Parkinson versus control. And I'm just showing you what we saw was significantly different between the groups, and the training set of subjects for the human microRNA. But when we actually combine the human microRNA, the piRNA, and the microbial content and train the best model possible, we can see performance that is indicated by the plot on the right of this slide. So more than 90% accuracy is achievable with as few as really a half dozen important molecular measurements and ratios between some of those. So we're very excited about the potential utility of saliva to be potentially diagnostic for Parkinson's at its earliest stages. Finally, moving on to COVID-19, looking at infectious disease, this was something that we were excited about the opportunity because there was a need. There was clearly a need a few years ago at the beginning of the pandemic to have a better assay. We knew that saliva could inform us about the microbiome content based on the studies we had already done to date. So all that we really needed to do was use the same swabs we were familiar with, design a better PCR assay, determine how low the limit of detection was for the virus, show that it was useful and specific and sensitive, show that the salivary content of the virus would actually be stable across transport, show that that was also robust to interfering substances that could appear in the mouth. Along the way, we decided to also develop a pooled testing methodology, and we achieved an FDA EUA for both the individual and the pooled testing. The pooled testing methodology is illustrated on the right of this slide. Essentially, we were able to have up to 12 subjects, swabs, the content pooled together, and then examine that one pooled sample. If the pool was negative, it was a clinical negative result. But if the pool was positive, the individual swabs would be re-extracted so we could determine which one of those samples or multiple samples within the pool were actually positive for the SARS-CoV-2 virus. The sensitivity that we had for the individual test and the pooled test both achieved the best in class. Another very important aspect of this particular swab was that the stabilization that it provided for the viral RNA allowed us to optimize genome sequencing protocols. And the last thing that we were able to add as well, because of the stability of the RNA in these swab devices, we could also look at host responses to the infection. So I'll just hit some of those highlights. This is an example of our weekly testing volume from when we received the authorization to begin clinical testing. And you can see that many weeks we were doing in excess of 80,000 tests from our single upstate New York laboratory locations. The positive testing results had a slightly different time course in terms of their peak. If you look at the summary of where most of the positives were, you can see from this map that we were derived most of those samples from the central New York area, but there were also plenty of samples coming in, thousands of positives from the capital region of New York, from Western New York, from Northern New York, from the Long Island, New York City area as well. All in all, we had about 30,000 positives that we were able to submit for sequencing. Through the sequencing, we were able to determine which specific lineages were showing up in different parts of the state across time. And that's what you're seeing displayed here in this graphic. There is a clear expansion of each one of the major waves of the lineages. They would become dominant. You just saw all of the lineages. Now here's the Delta. You can see the Delta outbreak. And now we'll look at the Omicron outbreak. And that includes BA1 and then the later BA derivative strains. Now, more important than just looking at the geographical location of where these strains were, was examining the sequences of the virus for the number of mutations. because the mutation rate could affect the sensitivity of PCR tests. It could affect antibody responses. It could affect the spread of the disease. And surprisingly, one of the things we found was that there were a large number of mutations that had accumulated in the viral lineages, particularly during this period when BA1 was expanding. We were gaining one new mutation approximately every week, and that was about twice as fast as what we saw before BA1, so in the phase leading up and including Delta. And it's leveled off since then. Now, when I saw this, it reminded me of the movie Jurassic Park. And you may recall, if you've seen the original movie, that there's a scene there where the character played by Jeff Goldblum asks the question of the scientists, are you concerned about the dinosaurs taking over the island? And he's reassured by the scientists that those dinosaurs could never do that because they're all female. And his response was, life finds a way. So I would suggest that this virus we've all come to know so well is very much like the velociraptors and has taken over the island. One of the unexpected things from our studies of the viral mutations and tracking these lineages that we've just uncovered is that, in fact, the more evolved the virus is, actually, it appears that there is a increase in the viral load. So what you're seeing here plotted is the number of cycles to threshold for detection of the virus. So indeed, life is finding a way. This virus is mutating more. The viral load is getting higher. It's showing up more rapidly now than it ever has before. The last thing I'll point out that we've looked at in terms of the SARS-CoV-2 is actually looking at the host mRNA responses. And we've done this in samples of males and females who were vaccinated or unvaccinated, who had every major lineage that we encountered. So this is more than 250 subjects. All of these use that same RNA sample that was collected for clinical testing, that was used for sequencing of the virus, and now is being used in this example for profiling the host response. Because we want to know, potentially, are there differences in what you're doing about these more evolved strains of the virus? Are you getting rid of it sooner, or are you worse off now that the virus has changed its sequence so much? Now, I won't go into the details of the differences that we're seeing between strains, but I will point out that when we look at how each of the lineages respond in common and look at the functional enrichment of the gene networks that are most significantly enriched, we see absolutely a profound defense response, an immune system, a charge against the virus is being mounted. So we clearly see that the virus is being attacked by the host in many ways, and we can actually track this in different levels, in different persons, in different lineages. Just to provide one more example, how the effect of vaccines come into play and what we can detect. This is an example of a gene expression profile for approximately a dozen genes, and we're looking at how they respond to viral infection, and that would be the blue line, compared to just vaccination, which is the orange line, and how they respond in persons who had a breakthrough infection, which is the gray line. So all of these genes, this whole panel of about a dozen genes, shows a robust viral specific response. They don't change from just vaccination, and they have a blunted response in people who had a vaccine before getting infected. So these are the types of things that we're learning that we can uncover from saliva. And just to summarize, saliva-based multi-omic analysis has revealed consistent changes in human and non-human RNA molecules that appear clearly to aid in the development of tools that could advance early diagnosis for brain disorders or infections. And rapid identification and full genome sequencing of microbial and viral genomes is certainly easily accomplished alongside the profiling of the host responses in those samples when the RNA in saliva is well preserved. And this has enabled us to monitor emerging variants of concern, as well as the people and the responses to those. I just want to thank the many colleagues who have participated in all of this work along the way, especially Stephen Hicks, Dragos Mihalia, Susan Baser, Andy Berglund, and Mr. Richard Ziwig, who is the founder and CEO of Quadrant, and countless others. And I'm happy to entertain any questions that you may have, which you could submit to the chat or after this session to my email. Thank you for your attention.
All right. Hello, everyone. I'm Dr. Forrest Wright from Quadrant Laboratories. That's the clinical and research arm of Quadrant Biosciences. I'm happy to be here today to discuss some of the foundational work that has gone along hand in hand with some of the cutting edge and saliva-based discoveries from our academic collaborators, such as Dr. Frank Middleton, as well as the directions we're moving in to develop clinical diagnostic tests for a number of neurological and infectious conditions. Just a quick note on our company's mission here at Quadrant. We use the strengths of our academic and medical collaborators, as well as our own discoveries, and create highly accurate diagnostic assays that improve access for patients by developing novel molecular assays with rigor, using saliva as a source material, and bring them through the commercialization process to address both clinical and public health needs. So, what do we do with all of this novel technical research and all this high-level collaborative discovery? Typically, our clinical research team is out there obtaining research samples in sufficient volumes, often not only in the hundreds, thousands, potentially even tens of thousands of samples. We use these samples to then drive our own internal novel discovery by identifying these differentially expressed molecules such as small RNAs, microRNAs, Pee-wee interacting RNAs, and then also confirm the research findings of ourselves as well as others. From this point, we use commercially available diagnostic software to make a diagnostic call if it's available. Often with this kind of novel research, it's not available yet. So in that case, we have a dedicated analytics team that can use these data in a machine learning setting to develop novel disease or condition-specific diagnostic tools. Our analytics team is in various stages of development with several of these novel tools. And additionally, we perform, on top of this new machine learning tool and diagnostic condition-specific algorithms that we're developing, we perform the typical translational developmental work that prepares us for regulatory applications, such as improving our assay throughput, identifying and validating our orthogonal detection technologies, and determine our operating conditions, generate our regulatory documents, such as SOPs or IFUs. Some examples of this development work are seen here. We ensure robust detection of microRNAs across the spectrum of sample qualities, and here indicated by the RNA integrity number, or RIN score. This is a common measure of RNA qualities. As you can see, detection of and quantitation of these molecules is consistent in replicant samples of various qualities, even in lower abundance microRNAs, such as the one shown on the left. Further, confirmation of molecules of interest for diagnostic use is necessary using at least one orthogonal technology, here shown by qPCR. Several microRNAs and small nucleolar RNAs are measured across 48 independent samples, and these show consistent detection by qPCR, which indicates consistent expression across the individuals tested. When comparing this to our next generation sequencing work and the detection using our Illumina sequencer on the right, this trend also holds true, and that indicates a reasonably strong correlation between the two detection modes. So all from saliva, we can do all of this. Saliva-based sampling allows development of all of our current products in our pipeline. And these vary in the use of technologies that include Sanger sequencing, next generation sequencing technologies, and qPCR, just to name a few. And a single swab can provide us with everything from genomic DNA, large and small RNAs, as well as microbial and viral genomic materials, be they DNA or RNA. Of our current products, I just want to take a moment to highlight the development of just a few of them. Here, I'll briefly discuss diagnostic assays for autism using both the genetic and epigenetic tools, a traumatic brain injury epigenetic test, and an improved SARS-CoV-2 test that includes the addition of several other common respiratory viral pathogens. As indicated previously, autism is a highly heterogeneous source of causes, and the current modes of testing include genetic panels. However, genetic testing can account for potentially up to 10 to 20%, so maybe 1 in 5 cases. And our two assays will leverage the ability of our lab and the use of these saliva swabs to obtain genomic DNA as well as total RNA from saliva to perform both whole exome sequencing and small RNA sequencing to broaden the scope of ASD identification and diagnosis, as well as potentially allow us to elucidate some of the epigenetic causes. We intend for this product to significantly reduce the timeline from when a neurodevelopmental issue may be first identified to a proper diagnosis, which currently can be on the order of years for many children. This allows for a much earlier application of critical therapeutic interventions and gives us an opportunity to meet a much needed public health change. So not only will we be able to use this technology to diagnose developmental issues such as autism, but also for such things as traumatic brain injury or concussion. In the case of traumatic brain injury, there's a poorly understood risk of long-term symptoms. And we aim to be able to identify patients through our epigenetic technologies who are at higher risk for developing these long-term symptoms and allow physicians to identify and quantify this risk very quickly, apply therapeutics, get patients into specialized sources for treatment. Alongside the myriad of sequencing-based applications that I've discussed briefly so far, Quadrant has also shown that saliva-based materials can be used to diagnose acute infections, such as with SARS-CoV-2, as well as track the evolution of variants of concern. In this case, a respiratory pathogen viral panel is being developed to allow for detection of multiple common respiratory diseases simultaneously through multiplexing, and ensuring the proper kinds of sensitivity, specificity, and lack of cross-reactivity that we would expect in any other kind of diagnostic tool. So in all, just to tie things together a bit, we have a spectrum of things that we can do with these saliva swabs. We can do genomic, whole genome, whole exome sequencing. We can take a look at novel tools such as these epigenetic detection modes through microRNAs, PE-inactivating RNAs, and other small non-coding RNAs to do novel diagnostics in areas that have not either had epigenetic diagnostics before or really have a huge gap for diagnosis. And we can also attack immediate public health needs through things like COVID testing and other respiratory diseases, which if you're a parent, your kids may be suffering from right now. I certainly hope not. But, so with that, I'd just like to take a moment to highlight some of the teams that continue to contribute significantly to the work that you've seen and heard about today. I'll be happy to take any questions that you may have at question time, and thank you very much for your attention.
Thank you, Dr. Middleton and Dr. Wright. Next, I'd like to introduce Dr. Andrew Spiers, who is head of clinical innovation at Ziwig. As a medical practitioner, he has a passion for putting patients at the heart of innovative medical breakthroughs. Before following a path in medicine, his initial studies focused on mathematics, statistics, and business. Coming from a broad background in general medicine, he spent 2 years working in a specialist gynecology and obstetrics department, first at the Angers University Hospital in France, and then in the North Vaudois Hospital in Switzerland. Medical research has always been an important part in his medical practice, and he has contributed to over 50 publications on a broad range of topics, especially those related to the health and well-being of women. His mission at Ziwig is to bridge the gap between scientific theory and medical practice by bringing innovative medicine to the service of patients.
Good day, everyone. My name's Andrew, and I'm very happy to be here today to speak about microRNA and testing for endometriosis. As a brief introduction, let me talk a little bit about endometriosis, which is an incredibly complex disease. Endometriosis affects about 5 to 10% of women of reproductive age globally. This represents a population of about 190 million worldwide. Endometriosis is actually defined as endometrial-like tissue, which is found outside of the uterine cavity. It's characterized mainly by symptoms of pain and infertility. 3 subtypes have actually been recognized in current literature. We speak about peritoneal endometriosis, deep endometriosis, and what we call endometrioma, which would be like cysts which could be found on the ovaries of a patient. When one speaks about endometriosis, there are 3 main figures which come to mind. The first is 7 years. Endometriosis has a 7-year average time to diagnosis. That is to say, from the time when a patient starts feeling symptoms till diagnosis, there's a time lapse of 7 years in which patients will have numerous consultations. As you can see from the second figure, 20% of patients will have between 5 and 9 consultations, and another 20% will actually have more than 10 consultations. And finally, the third figure is 1 in 4, 25% of patients will actually have to undergo diagnostic laparoscopy in order for a diagnosis to be reached. During this time frame, patients will undergo a numerous testing, medical wondering, suffering, there's investigations, and often a big discrepancy between symptoms and clinical presentation. A study which came out in 2021 by Meismer et al. Actually showed how endometriosis will affect a patient through their adolescence and adult life. And symptoms are highly acute during critical phases over a patient's lifetime when they will be making decisions about education, about family life, and their professional environment. So this brings us to what are the tools which a practitioner would have at their disposal in order to diagnose endometriosis. And this table briefly outlines the different approaches which a practitioner could take. Firstly, there's medical questionnaires. And although it's very useful to take a detailed patient history, unfortunately, the specificity of these tables and these questionnaires is very low. Actually, the European guidelines from the ESHRE came out in 2022, and they showed that there is no evidence that questionnaires actually reduce the time to diagnosis. The cornerstone of current guidelines and approach to diagnosing endometriosis is actually medical imaging. But as we know from the European Society of Human Reproduction and Embryology, that even if imaging does not find endometriosis, this does not exclude the presence of the disease. And this is notably for the peritoneal form of the disease. So, as we can see, low accuracy for early stage lesions with imaging testing. And this will bring us to blood work. And blood work, as you can see, has low sensitivity and specificity. And finally, we have surgery, which is an option which does have a fantastic sensitivity and specificity and is often recognized today as being the gold standard in complex cases. However, guidelines have recently stated that even in the presence of negative histology, one cannot entirely rule out the presence of disease. The saliva test developed by Ziwig will actually provide fantastic information to practitioners by giving a highly sensitive and a highly specific diagnosis. The technology behind the Ziwig endo test is based on the analysis of microRNA. Many of you will be far more experienced than I am in the field of gene expression, but I'll very briefly take you through the role of microRNA. So microRNA are single-strand nucleotides which are from the transcription of DNA. Their primary role is to link to messenger RNA and thereby silence the production of proteins. Much study has actually recently been done in the field of microRNA. And in 2018, a very high quality study by Panier et al. Actually looked into the different pathophysiological mechanisms and links between endometriosis and microRNA. And we can see that they actually found specific microRNA that were linked to cellular survival and proliferation, to inflammation, to angiogenesis, as well as other pathophysiological pathways. Ziwig has been highly active in medical research, and we actually designed the endo test through a very logical process. First of all, we validated the machine learning algorithms and artificial analysis. We did this by looking at a very large database of information, mainly clinical information, provided by patients. And from this, we were able to determine that machine learning was able to crunch numbers and look at far greater quantity of data than any of our previously looked into statistical models were able to do so. We actually then took this a step further in a second study and started looking at the pathophysiological pathways linking microRNA to endometriosis. A third study we did then actually looked at the full human transcriptome of microRNA using next generation sequencing and how interactions with endometriosis could be described. From there, we actually were able to produce, firstly, a blood-based microRNA signature. Now, this signature was based on 86 different microRNA and produced outstanding diagnostic results with an area under the curve, which was at 98%. We initially thought that we would have to bring together information from patient questionnaires and do initial salivary work, patient questionnaires, so that would be a non-invasive way of looking at things, and then from there do after that screening, do a blood-based microRNA test. So we did not expect the fantastic results that we actually had from the salivary signature study. The salivary signature is based on 109 microRNAs and, as I'll tell you, has outstanding accuracy. Actually, there's a 98% area under the curve. And this means that the signature is actually able to stand alone as a diagnostic tool to diagnose endometriosis. We then actually took our analysis of microRNA a step further, and we looked at how different phenotypes of endometriosis patients could be impacted and described using microRNA. And we were able to determine and distinguish between two populations: patients who had endometriosis and suffered from infertility, and patients who have endometriosis and who do not suffer from infertility. And this is our latest breakthrough in terms of the information that we're able to draw from microRNA in the field of endometriosis. This slide shows the complexity of the microRNA expression. On the left-hand side, you can see that there's a diagram of a single pathophysiological pathway, and you can see all the microRNA that are involved in this pathway with different functions such as apoptosis and insulin regulation functions. We looked at, during these studies, the full transcriptome, over 2,600 microRNA biomarkers, which had hugely heterogeneous functions. And these led to, first of all, the blood-based signature, which, as I mentioned, was based on 86 microRNA biomarkers with a fantastic specificity and sensitivity and an overall diagnostic accuracy of 98%. On the left-hand side, you can see the relative importance of each microRNA contributing towards this model. An interesting side note is that even those with the slightest importance in the overall model are very necessary to the performance of the model. If one were to take out 5 or 10 of them, the diagnostic accuracy would actually be influenced far more than one would imagine. So this actually shows the huge complexity of the model, which isn't simply a sum of different microRNAs to reach a diagnosis, but it's a combination of how these microRNAs are interacting and the artificial intelligence model, which is able to map this and reach a reliable diagnosis. The salivary microRNA signature is based on 109 biomarkers and again has outstanding accuracy with an area under the curve of over 98%. Again, the full human transcriptome was looked at, over 2,600 microRNA, to design this model. And the saliva model is actually able to pick up endometriosis at all stages of the disease. Our cohort included not only early and, or I'd say not only late stage disease, which as we know is easier to detect on imagery, or even during a diagnostic laparoscopy, but our test, or the endo test, is able to pick up early stage disease. And during the trial, about half of the patients showed stage 1 and 2 disease, and the other half stage 3 and 4 disease. This is a major breakthrough for patient treatment because, as we know, catching the disease earlier will also allow for not only patients to have better quality of life through earlier treatment, but also it will allow for treatment specific to fertility. And endometriosis has 2 long-term consequences. The first would be that once chronic pain has set in, it's very difficult to break the cycle. So the earlier you're finding the disease, the less chance there is of chronic pain setting in. And the second thing we know is that chronic inflammation and fibrosis does have an impact on fertility. And if we're catching the disease earlier and reducing these symptoms, we can hope to have a very favorable impact on fertility. This slide actually shows what the endo test itself looks like. So, up. It's a simple collection kit, which is designed by DNA Genotek. We're very happy to actually be working with leaders in the field. And patients will simply provide a saliva sample, and there's a liquid in the capsule that will preserve the test. We know that saliva has huge advantages over blood, not only in terms of the fact that there are more, I would say, a higher variety of microRNA found in saliva than blood, but that the microRNA are actually more stable. This means that we can test samples that have been provided over 30 days earlier and still find outstanding results. As a conclusion, the salivary microRNA endometriosis diagnostic test has a fantastic accuracy, a sensitivity and a specificity, which will allow for it to provide a clear answer to practitioners. It's safe, non-invasive, and as I mentioned, in a stable format. This should definitely contribute towards a decrease in medical wandering. That is to say, a decrease also in the 7-year delay during which patients are suffering from the symptoms of the disease. And overall, it will reduce the burden on our healthcare system.
Thank you to our presenters today, Dr. Middleton, Dr. Wright, and Dr. Spiers. Now we have a few minutes for some questions. Thank you for the participants. You guys sent in a number of questions. So let me get started. We only have a few minutes. First question, maybe to Dr. Spiers. Is there any evidence on the reduction of unnecessary surgical procedures due to earlier diagnosis with the salivary endo test?
This is actually a fantastic question because it's one of the primary objectives that we have as for the endo test. And to be honest, we would like for the endo test to replace diagnostic laparoscopy. Because after imagery, if the imagery finds clearly endometriosis, then the diagnosis is done, in which case. But if the imagery is normal, or if there's a doubt, then instead of going to diagnostic laparoscopy, this would be the time when the endo test could be done and provide an answer without laparoscopy and all the risks associated with it being necessary.
Thank you.
Thank you for this question.
Next question to Dr. Middleton, maybe, or Dr. Wright. How did you give a diagnosis of autism to the patients? As you said, it is difficult to base it on questionnaire alone. So how did you diagnose autism patients to confirm the saliva-based test was more effective?
So that's an excellent question. What you saw summarized today was the results of research that's been done in a clinical setting, but this did not by itself render a diagnosis. Only a healthcare provider can make a diagnosis. So the information from the saliva test was provided to the healthcare provider, and there's a whole report form that's generated which explains those results and the statistics around it so that they can understand it when they communicate with the parent.
Thank you. There was a question whether any RNA-based biomarkers have been reported in saliva for cancers. So maybe I'll take that. This wasn't part of these two talks, or these 3 talks. I know that in literature there is indication that there are biomarkers for oral and even systemic ones, lung, pancreatic, and others. I think what the talks today did show was that, we're looking at exosomes, microRNA that are not originating in saliva. So we could probably extrapolate that there are potential other biomarkers in saliva as well. Next question. I guess it's very related, Dr. Middleton. You described the transport of these markers across nerves from brain to saliva. Do you have any indication that EVs can pass the brain-blood barrier and be transported to saliva?
In the other direction, going back to
I think into saliva, but through other means other than the nerves.
Yes. So there's what's called a glymphatic system. So there's a drainage system around the meninges that collects the waste products and other products. Could include proteins, it could include microRNAs, could include extracellular vesicles in the major lymph nodes, ultimately. And so there's a conduit as well that we believe could provide an opportunity for transport. It doesn't have to be coming through the nerve. It could be in the perineural space as well, which is related to this glymphatic system. So there are a lot of potential ways for things to get out into the saliva, but I would contend that that's an easy path because that's the path rabies virus takes. For example, when someone has an infection, a massive rabies infection in the CNS, it easily transports out to the salivary glands and saliva.
Thank you. Dr. Spiers, another one for you. Did this saliva test for non-specific endometriosis or subtype-specific cases? And if it is non-specific, is your hope to develop a subtype-specific test, or is this test part of a triangulation approach for diagnosis?
Again, a very good question. So the endo test at this point will provide a yes or no answer. Does a patient have endometriosis? It will not go into the details of different phenotypes. This is something that we're looking into because as we move forward with our research, we realize that there is a huge quantity of information available in microRNA. As I mentioned, the most recent study we've done has found a phenotype which is patients with endometriosis who suffer from infertility versus those who have endometriosis and are not infertile. And we'll be moving forward on this research to find if we can distinguish between peritoneal types of endometriosis, deep type endometriosis, and endometrioma. A second part of the answer, which I'll very briefly get into, is that the endo test, you mentioned triangulation, is not meant to replace imaging, which would be complementary, because once endometriosis, especially late-stage endometriosis, is found, it's important to map these lesions.
Great. Thank you very much. Maybe one last question. I know we're at time. The panelists will have asked these questions and can reply to them after. You also, everyone here has access to the emails in the chat, so please feel free to reach out as well. So last question, maybe Dr. Wright or anyone on the panel here. Could we miss something in diagnosis due to unstable nature of RNA if very low levels are detected? So it's like an assay question here.
So certainly it depends on the nature of the molecules that you're taking a look at. With much of our epigenetic work, we're actually pretty fortunate that microRNAs in particular seem to have a pretty lengthy stability compared to your ribosomal RNAs or your much more commonly studied longer RNAs. And kind of everybody understands that RNA is a lot less stable than DNA. But actually, one of the figures I showed in my slides is taking a look at some samples that were all across the spectrum of qualities and degradation states, and our microRNA responses and counts in our sequencing data were robust, even in what are generally considered to be low quality samples, ones that would be thrown out by other core facilities if you were looking at other kinds of larger RNAs.
Thank you. Any other comments on that last question?
I think it does depend on the nature of the RNA that you're trying to study and the nature of the condition that you're trying to study. So there are definitely going to be organs that may not contribute highly to what you could detect in saliva at this point. But the audience has heard examples of some organs from the speakers today that clearly do. So it will depend on the nature of the question.
Great. Thank you. And with that, we'll wrap up the Q&A session.
Okay, thank you very much. I thought the talks and the data that was being presented was so impressive, and thank you, Rafal, as well for chairing the Q&A there. You did a wonderful job. That is all we have time for today. So I've already thanked our speakers. Thank you to you for joining us at this webinar. I'd also like to thank our sponsor, DNA Genotek. Don't forget, this webinar is available on demand, so you can watch it back at any time. Just check for the link in your email. I hope you enjoyed that. Thanks again. Have a lovely rest of the day, everyone. Bye.
Thank you.
Bye-bye.